Discover ASAP, the video interview series where we highlight the top findings and tools connected to the ASAP network. Today I'm talking with Zac Caffall, a Senior Research Analyst from Team Calakos. For today's episode, we're going to be discussing the Integrated Stress Response Reporter Spotlight developed by Team Calakos. Zac, can you get us started by telling us briefly about your team's ASAP project and how this tool is being used in the project?
Well, when we first developed the Spotlight Reporter, it helped us discover that striatal cholinergic interneurons use a stress pathway in an unusual way. Basically, these cells activate the integrated stress response, or the ISR, at steady state in healthy brains. We also found that if we inhibited this pathway in those cells, that there were differences in dopamine signaling to cholinergic interneurons and effects on motor skill learning. Our ASAP grant follows on these insights by understanding the significance of the ISR pathway in cholinergic interneurons in Parkinson's disease. So we are using the Spotlight Reporter to understand just how far and wide this property of striatal cholinergic interneurons extends. We're asking, is the level of the ISR activity in striatal cholinergic interneurons disrupted in Parkinson's models? And do other cholinergic neurons outside of the striatum show this tonic activation of the ISR? Are there regional, sex, age-dependent differences among striatal neurons? And are there other types of neuromodulatory neurons besides cholinergics that rely on the ISR for normal steady-state function?
Great. I think this is really interesting and such a useful tool. So I want to start off by high level talking about exactly how the Spotlight Reporter works and how you use it to identify the integrated stress response in cell types.
Sure. So when stress activates the ISR, there are major shifts in which of the messenger RNAs are being translated into protein. So most of the time with the ISR activation, general translation shuts down, and a subset of a few, some may think up to a third of the proteins are actually being either maintained at a steady state or increase their translation. And this largely happens because of a shift in the kinetics of protein synthesis, initiation by a complex called eIF2, so resulting in a different open reading frame being used. So we put GFP, or green fluorescent protein, into the reading frame that is read when the ISR is low or not active. And we put red fluorescent protein, we call it tandem dimer tomato because it's red, in the reading frame that will express it when the ISR is high. So at the high level, green is ISR off and red is ISR on. So Spotlight, this reporter, provides a readout of the functional state of protein synthesis, initiation, and translation that is regulated by eIF2 initiation complex. It does it by using regulatory upstream open reading frames that were described mostly by Ronald Weck for the best described effector of the ISR, which is a transcription factor called activating transcription factor 4, or ATF4. So the 5' untranslated region of the mRNA has several upstream open reading frames, basically a start codon, a stop codon, and another start codon, and a stop codon, and depending on how depending on the rate of translational reinitiation after these upstream open reading frames, you get selection for particular reading frames. And in this case, this is a regulatory system that has overlapping reading frames on the same mRNA for ATF4. So when you have low ISR stress, you actually have a nonsense protein that is translated over the ATF4 reading frame. So, however, when you have high ISR, what happens is that nonsense upstream open reading frame is skipped, and you get translational initiation starting for ATF4 itself. So for the spotlight, we basically replace the nonsense initiation, the nonsense peptide upstream open reading frame with green fluorescent protein, and then the ATF4 with red fluorescent protein. So typically with no stress, we get a cell that is infected and it's green. And then when you add stress, it now stops making green and starts making red.
Great. That's a really great description of, I think, a very, complex and elegant system you guys have described. So I think one of the big questions that I think anyone who is watching this video is going to have is, how do I use this reporter in an experiment? And I know you guys have sort of two versions of this. One is a viral a plasmid, and the other is that you have a knock-in mouse line. And so hoping you can briefly talk about delivery of the plasmid for use in an experiment, and if there's any critical steps to this process that you think that people should be aware of.
So I'm gonna follow along here, but so Spotlight is kind of a unique in that it provides this functional readout of ISR on protein synthesis. In a way, the red to green ratio can be thought of as a thermometer to help read out how active the ISR has been in the cells. And these kinetics of the readout relate to the half-lives of these fluorescent reporters. So they typically over 24 to 48 hour time period, you'll see the true signal. So we have found that most cells are green, and highly red cells do stand out and can draw your attention to new biology in a way that may not be as easy with conventional IHC. So you can do IHC for the individual components of the integrated stress response. And though many of these antibodies have low signal to noise and poor specificity, so phospho-eIF2α antibodies are most widely used. And we've posted an optimized IHC protocol for that that we collaboratively developed with other ASAP investigators. But depending on the application, phospho-eIF2α staining may be too transient or weak. So with the Spotlight mouse line and its Cree dependence, it's a really nice tool to light up particular cell types and survey it for variation in ISR activation. So we do have the constitutive Spotlight AAV backbone. That's already available through AdGene, but we've made available the Cree-dependent Dio Spotlight viral backbone through the ASAP hub. And we are in the final stages of work of validating our Cree-dependent knock-in mouse, which is at the Rosa locus. And I just as a hint of what it's looking like, this image behind me is the hippocampus from a mouse from our Dio Spotlight mouse that was crossed with a nesting Cree. So you can see it's really intriguing to start to see which cells are going to start lighting up. The one thing about the difference about the Dio Spotlight mouse here is that we don't have the problems with viruses of having multiples of infection and varying expression levels. So that really drew us to wanting, especially when we're looking at red hot cells in a brain after a viral infection, they could have just gotten many more copies of the virus. So viral tropism was a confound for that. This hopefully will surpass that or make it not necessary to be concerned about.
Great, that's wonderful, and that's such a beautiful image behind you. I think anybody who's done any cell tagging can really appreciate that image. So You talked about the advantages that the knock-in mouse line has over the AAV, but I just want to briefly touch again on if someone was choosing to use the AAV, how you recommend administering that to the animal, and if there's any tips or tricks you have for improving or increasing viral transduction.
So it's a AAV2 backbone, so it could be packaged into any AAV envelope that a user might want, either for like one of the PHBEBs for peripheral delivery. That's what we have done that and given intravenous injection of it, and that's what we use for publishing our the science paper a few years back. But typically it can be packaged just like any other virus. The main thing that we've had concerns about is whether or not we're looking at, if you're looking just at one fluorophore or the other fluorophore, we're really finding that it's the ratio between these that is going to give us the most information. And especially as we're characterizing the mouse, it's while there are trends that are pretty obvious when you look at the individuals, it's more important to look at the ratio between them, especially since we were looking within cell. And typically we do use IHC to amplify it, and especially in the mouse. You might not need it as much in the virus if you're getting multiple copies of infection. So, using the virus if you're doing if you're high titers of virus, but in the mouse we tend to use IHC to amplify the RFP and the GFP signal together.
Great, that's really good to know. And are there any other special considerations for processing the tissue with spotlight fluorescence, other than knowing that you really should amplify the signal with IHC?
The one step that we developed with another group at through ASAP was the use of alkaline retrieval, and that's part of our protocol online, I think.
Great, that's good to know. And I was going to ask you too about any special considerations for analyzing tissue with spotlight fluorescence. I think you've already touched on maybe the most important thing, which is that it's looking at the ratio of the green and the red signal to each other. But are there any other things that people should consider when they're working with spotlight tissue and trying to draw some conclusions from the signals that they see?
I think as we're surveying certain cell types in this mouse, it's picking your background correctly, figuring out which cells you're going to be doing the direct comparison with, because there's so many different cell types. The idea is always best to try to pick a region. There might be regional differences. We don't know yet. We're working on that. So whether or not if we're looking at cholinergic interneurons outside the striatum in the brainstem, in the spinal column, are the basal levels of all the cells there higher? Basically, are your comparisons going to be one cell versus all the cells in the brain, or one cell directly within the region itself? So we try to get adjacent cells as our background, as our comparator for when we're looking at particular cell subtypes. Theoretically, depending on which Cree you're using, you should have recombination in all certain subsets. So we're using a nesting Cree at the moment. This is a nesting Cree mouse behind us, and all of the caveats that go into using individual Cree lines will basically dictate with the sort of characterization controls that you're going to be needing to use with this mouse. So I don't know if that answered the question. Hopefully it got somewhere.
Yeah, I think those are some useful tips, and I think that we will all eagerly await the characterization of the mouse line. I think that will be incredibly useful to everyone who's interested in studying the integrated stress response. Thank you so much for taking the time to talk with me today, Zac.
Oh, thank you for having me. It's been a joy.